Okay, Dana. So let's talk about the cloning strategy that you use for the IgNAR gene. Just the highlights.
The highlights. Okay, I want to clone this IgNAR gene. So to make this, first I need a vector. A vector is a genome where I can put or insert my gene of interest, and depending on what I want, I can clone it or I can express it. In this case, I want to clone it, so I choose a vector for cloning. And what I choose is a Pyes 2 HTH, and is a HTH because of its histidine tag. It's an special histidine tag that is from 10 histidines. So it can help us purify this protein. Well, we also need, there's not only like choosing the vector, we need to use like all the things the vector needs to have, for example, the promoter and all the antibiotic resistance, the promoters and all that. For example, the promoter, it's GAL1 that I can, with this I can control the expression. I can induce it or I can repress it. I can induce it with galactose and I can repress it with, I use galactose and I can repress it with glucose. Yes.
Great.
Also, at the resistance of antibiotic, this is to separate like my transform cells from the non-transforms. And my vector has the ampicillin resistance, so I can I can grow my cells in ampicillin resistant medium, so they only grow the ones that were transformed.
Okay, so just a thing, something. You mentioned that you use a cloning vector.
Yes.
What's the difference between a cloning vector and an expression vector?
A cloning vector is to make, I sorry if I make it, I say the same word, but I want to like replicate or like to the gene of interest. And if I want the expression vector, is that I want to like the gene or the protein, I want to express it in the host. It needs to be like expressed. For example, I don't know.
Okay. Maybe what you want to say is that the cloning vector, it's for that you want to have a lot of copies of the plasmid and the gene.
Yes.
And the expression vector, it's because you want to express the protein. Well, the gene to a protein.
Yes.
Okay. So maybe you want to reconsider that the vector that you use, it's a cloning or an expression vector. You want to have
the one the one I use is for expression. I confused.
Okay. Great. I know that you're confused because you tell me everything about the expression vector. Okay. So now that we're on the same page, what happens in a multi-cloning site?
Where in this site we have a lot of enzymes. These are restriction enzymes, and this is where I can insert my DNA insert.
Okay.
Yes, there is where I can put in my insert. Is a place.
Okay. So what does this insert has to have, and what does the multi-cloning site in your vector has to have to clone it in terms of enzymes?
I need the restriction enzymes in the vector.
Okay.
But these restriction enzymes, they don't have to be in my insert gene because it's not.
Exactly.
The work of the restriction enzyme is to cut the vector.
Okay.
I can put there my gene of interest. So if I need to make a digestion of both of my gene and my restriction enzyme. And if I make the digestion in the gene, they're going to cut it, and we don't want that. So we need also the primers
Okay.
on the insert gene. So when the digestion, we can make these sticky ends, so they can,
Okay.
where it's the same part they cut on the plasmid. So ligation with the ligase.
Okay, great, Anna. We can stop the recording. We already finished your interview.
Excellent. Okay.